Welcome everyone to this week's edition of the Extracellular Vesicle Club. This EV Club is being sponsored by the Extracellular Vesicles in Nervous System Special Interest Group of ICEV. And this group has had several activities already. They're also planning a meeting at the end of October, so be on the lookout for that. I think the registration is about to open as well. But this week we're going to have a presentation that is being moderated by YoungGyun Park from Korea. And so, YoungGyun, I'd like to hand this over to you now to introduce our special speaker today. And I would just like to ask everybody to keep to put their questions and their comments in the chat box, and then YoungGyun can help with moderating those questions at the end of the presentation. So, thanks so much for moderating today, and I'd like to hand this over to you.
Thank you so much, Ken, for your introduction about myself. So my name is YoungGyun Park. I'm an assistant professor at Korea Advanced Institute of Science and Technology of South Korea. And I'm very happy to moderate this talk. This is the fourth EV Club sponsored by the EViNS Special Interest Group. This EV Club of EViNS is organized by Snea Ikezu, who is in this room, I believe, and Stefano Lucino. And I'm here as a moderator for today. So today, I'd like to introduce today's speaker, Ermioni Arvanitaki. She's a PhD student in the lab of George Garinis at the Institute of Molecular Biology and Biotechnology at the Foundation for Research and Technology Hellas of Greece. She will talk about the role of the EVs derived from microglia to the age-related neurodegeneration upon DNA damage, which is a very interesting topic and which is also based on her recent paper published in PNAS last year. So, Ermioni, stage is yours.
Thank you. Thank you for introducing me and for giving us the opportunity to present this work in the EV Club. My name is Ermioni. I'm a PhD student in the lab of George Garinis working on the impact of genomic instability in mammalian physiology and aging. My presentation today will be about how microglia that accumulate DNA damage, as you also said, you also pointed out, also accumulate DNA in their cytosol, package the cytoplasmic DNA fragments in extracellular vesicles, and target neurons, which leads to neurodegeneration in our mouse model. First of all, we know cells have adapted multiple pathways to counteract DNA damage and safeguard their genome. Accumulation of these DNA lesions can lead to cellular malfunction, cell death, or senescence. So it's very important for cells to keep these mechanisms going and for them to function properly. Our lab has previously, among other things, used mouse models lacking a nuclease, important for some of such repair mechanisms, ERCC1. ERCC1 participates mainly in nucleotide excision repair, but it also repairs interstrand crosslinks and some double-strand breaks. And both full knockout and tissue-specific knockout mouse models, that is adipose tissue and monocyte-derived macrophages, we've worked on before, have revealed a key role for DNA damage in inflammation. So our question for this work was to check whether DNA damage in tissue-resident macrophages this time would have an impact on the physiology of mice, given that macrophages are integral for both physiological and pathological inflammation. Tissue-resident macrophages are long-lived. They have a different ontogeny. They derive from the yolk sac, which is a bit different from bone marrow-derived macrophages. They are more short-lived, and they play important roles in maintaining organ homeostasis. So for this purpose, to ask what DNA damage in tissue-resident macrophages does, we crossed ERCC1 flocks to mice with CXCR1 free mice. CXCR1 is a receptor that is expressed in tissue-resident macrophages. So our transgenic mice have locked out only ERCC1 in this lineage. And our first results were striking. As you see here in the videos, 6-month-old wild-type mice are able to extend and shake their hind limbs when they are picked up by the base of their tails, while on the other hand, knockout mice are unable to do so by the age of 6 months, and at around 8 months, they bring their legs closer together, they develop kyphosis, and then are unable to maintain their balance when testing it on a rotor test. So naturally, we then turned our attention to brain-resident macrophages, microglia, and the impact microglia might have in brain homeostasis. Microglia are actually integral to achieve brain function, as they patrol the brain for any pathogenic or other insults and support communication with neighboring cell types. They also promote synaptic pruning, etc. So DNA damage is a hallmark of many neurodegenerative disorders, and vice versa. Mice with inborn defects in DNA repair show symptoms of neurodegeneration, ERCC1 knockout mice as well. And neuronal cell death in general is a result of the dysfunction of many cell types, including microglia. So could it be that this defect that we have in this mice, that microglia triggers the paralysis we observe? And if so, how does that happen exactly? We first checked the specificity of our knockout. Total ERCC1 protein levels appear to be similar in 3 different CNS regions that we checked: the cerebellum, the cortex, and the spinal cord. Among others were Purkinje cells, binding positive cells that we saw in our immunofluorescence studies, while on the other hand, microglia seem to have a reduction in the levels of ERCC1. We additionally observed no differences in the myelination of various other CNS regions, and of course, including the ones I mentioned before. And of course, when we checked for cell death, we found at the age of 6 to 8 months, we found increased percentages of tunnel positive cells. Tunnel is marker for cell death. We found significant increases of those cells in the cerebellum, particularly in the Purkinje and granule cell layers, calbindin and PAC6 positive cells, and in spinal cords. And considering neurons are not DNA repair deficient, but microglia are, these cells must be initiating this response somehow. We found several DNA damage response markers were increased in both microglia isolated by Percol. So we are working with tissue cultures that we take microglia from Percol purification, and also some tissue sections. And we have found an increased DNA damage response in these cells, and also, but on the other hand, new and positive cells remained healthy in terms of gamma H2EX in the presence of gamma H2EX. This further supports our thoughts that it all starts from microglia, because neurons do not have DNA damage. So then we asked whether microglia as immune cells would be in a different inflammatory state. What we found in our tissue cryosections when we stained for with IBA1, which is a marker specific for microglia, we found highly branched microglia cells in knockout mice. They had a higher number of interceptions and a larger soma, and also we found a higher capacity for antigen presentation, expression of MHC2 and CD86 antigen presentation molecules, all of which point to activated microglial cells. Next, we checked because type 1 interferon is important for neurodegenerative, many neurodegenerative diseases and in DNA damage inflammation as well. We checked for the presence of this response and the presence of the nucleic acids that generated, some of those at least, of DNA species. Immunofluorescence studies, again, in isolated and cryosection microglia, pointed us to double-stranded DNA released in the cytoplasm of microglial cells, as you can see here. And this dsDNA, double-stranded DNA, is also sensed by the cytoplasmic DNA sensor SIGAS. We further searched then for the dsDNA source and found single-stranded DNA ablation by protein transfection. Well, we first did our DNA treatment to check that dsDNA would disappear and the staining would drop. I'm not showing this here, but we also saw was a reduction in the ssDNA level. So we did a protein transfection with an S1 nuclease that also degrades ssDNA, and we saw dsDNA also being reduced. So one hypothesis for how this might be happening was that some repetitive sequences of single-stranded DNA might be reannealing to give rise to dsDNA. And there were also some previous reports on ATM-deficient microglia cells that accumulate DNA damage like ERCC1 deficient cells. And they did sequencing in the DNA, in the cytoplasmic DNA of the cells, and found that the majority of these repeats belong to satellite repeats, these GSAT-MM repeats. And we also checked and found something similar in our, there was a presence enrichment of this DNA in our cells. That all in turn, besides SIGAS, which we said about, also activates phospho-STING and triggers interferon beta production in the cerebrospinal fluid derived from microglia as well as other cell types. And I'm saying from microglia because when we culture these cells, microglia, and either it is knockout microglia or a topocyte-treated microglia that causes DNA breaks, we see an accumulation of interferon beta staining in the span of 6 hours. And what was also very interesting for us was that neurons that we previously found to be dying, like skull-binding cells, PAX6 cells, and spinal cord cells, appear to be sensitive to type 1 interferon signaling as well, meaning that they express type 1 interferon receptor if not. And besides being receptive to type 1 interferons, and the neuronal cells themselves were able to produce interferon beta. And also curiously, when we first stained our dsDNA and our brain sections for the presence of dsDNA, we found an increase in the levels of dsDNA in many of those neurons as well. So microglia and neurons accumulate cytoplasmic DNA. But since microglia is the source of DNA damage and the source of cytoplasmic DNA, how would it be possible? We wonder if the DNA could be transported from one cell to the next, from microglia to neuron. And that is how the potential presence of extracellular vesicles, EVs, caught our attention. Evie had done some work already in the lab before, so she was a bit familiar with that. EVs have been acknowledged as transporters of bioactive molecules in the brain, and activated microglia are EV producers, EV hyperproducers. So we hypothesized if EVs might be targeting neurons to dispose of their dsDNA cargo. To test this, we purified EVs from the brain parenchyma. We did several ultracentrifugation steps for the removal of cell debris, and larger microvesicles, and then we did super gradient centrifugation to enrich for smaller vesicles. Protein extracts derived from these fractions were rich in CD81 typical EV marker, CD11B microglial marker, which speaks of their origin, and the DNA damage response marker gametes 2EX. Particularly in the case of knockout EVs, gametes 2EX appeared to be enriched, which gave us the impression there might be some fragments there. We then collected these EVs and observed them in a scanning and transmission electron microscope, which gave us the impression knockout EVs were more electron dense, judging by their darker shade, so they could indeed contain nucleic acids. And when we lyse those cells, perform DNA extraction, and run it in a polyacrylamide gel, we saw that there were fragments just under 400 base pairs in length. So then, to check if knockout EVs can indeed be taken up by neuronal cells, we initially incubated a neuronal cell line as HSY cells with with knockout and wild type EVs that were labeled previously with exoflow and picogreen. Exoflow is an EV-specific dye, and picogreen is a dsDNA-specific dye. So what we found was a higher colocalization of picogreen signal with those cells in the case of knockout EVs when they were donors. And at the same time, we checked if something similar could happen in vivo. So we again labeled our exosomes, only knockout exosomes this time, with exoflow to be able to track them. We did an intranasal delivery in mice, and then we saw that there was a preferential uptake for knockout EVs in neurons, that's one positive cells, when compared to other cerebellum cells, for example, like CC1 oligodendrocyte or GFAP astrocytes. So to sum up what we've talked about for now, you have cytoplasmic DNA originating from microglia, which is triggering an interferon response. The DNA is packaged in EVs. You have EV biogenesis, and these EVs are able to target neurons and transport their dsDNA cargo there. But ultimately, these neurons die. So which one of these secretory phenotypes of microglia could be responsible for apoptosis? We then consider how it could be possible to answer this question, and the answer actually came from two-photon microscopy. The main advantage of multiphoton microscopy is that you can image samples that have greater tissue thickness when compared to confocal microscopy, for example. That means you can take your piece of tissue, 300 micrometer brain slice, and perform the treatments for a short duration of time, and then you can fix and image it. In our case, we tried treating the slices with wild type and knockout EVs, type 1 interferon, or both at the same time, and our readout was Purkinje cells. We picked Purkinje cells because they are, on the one hand, they are large and suitable for analysis, and on the other hand, they fit many of the criteria that we had for interferon receptor signaling, double-stranded DNA, and cell death. So as you can see, Purkinje cells are more susceptible to taking up knockout EVs, and that effect is pronounced when type 1 interferon signaling is involved. And the same goes for colocalization with the picogreen signal. Knockout EVs when knockout EVs are added, picogreen signal is colocalized more frequently with cal-binding Purkinje cells. And even more importantly, cell death, cleaved caspase 3 signal. And when we block type 1 interferon signaling with neutralizing antibody against IFNAR, we see a reduction in cleaved caspase 3 levels. So then how can you reverse all this? We wondered whether we could at least alleviate the inflammatory load in knockout brains, and that would mean we could delay the onset of neurodegeneration. For this reason, we developed a therapeutic strategy where engineered EVs mice would be treated with engineered EVs. We isolated the exosome from a fibroblast cell line, NIH cells, and decorated them with a custom peptide containing part of the CD63 peptide sequence. CD63 is an EV marker and part of CD11p, a microglia marker. So these fusion peptides decorated exosomes from the outside, and from the inside we added DNAs. But we first had to check that our exosomes would reach microglia, so we incubated our EVs with these peptide with this ligand or didn't incubate them, and so that when you add the ligand, the EVs can more efficiently reach microglia. And when you add the DNAs to the mix and treat cells, not intranasal delivery this time, and you treat cells that have dsDNA, etoposide-treated cells, you see a reduction in the levels of dsDNA. So EVs can reach microglia in the brain, and DNA levels can drop upon the addition of DNAs. So we now had to test if we could reduce neuroinflammation and neuronal cell death in knockout mice. We started treating both wild type and knockout mice at the age of 3 months, which is about 3 months before mice start showing signs of neurodegeneration. We again delivered EVs intranasally twice a week for a duration of 6 to 15 weeks. And this series of experiments was pretty exciting for us because knockout animals treated with DNase EVs, the line that you see here, was at the level of the wild type treatment, while knockout mice, the wild type mice, while knockout mice treated with naive EVs developed neurodegeneration earlier on. And when we wanted to check what was happening, so when we picked a time point earlier on at around 18 weeks, which is 1.5 months, 2 months after the initial treatment, where we see no difference in the rotor assessment, mice are able to still maintain their balance. We see a drop in the dsDNA levels in microglia. We see a drop in the MHC2-CD86 levels, which means microglia are presenting less antigens now, and they also produce less of type 1 interferon. And that was also accompanied by a smaller fraction of cells that stained positive for NXMPI, which means neurons now appear to be dying in mice treated with DNase EVs. So going back to our model, using our EV-based strategy and by targeting cytoplasmic DNA, we were able to ameliorate neuroinflammation in knockout brains and prolong the health of neurons. And with that, I would like to thank Evie and Katerina, with whom we have worked together in this project. Of course, Professor George Verinis, George Nyotis for his work in exosomes and many of the experiments that you saw. The previous master students in the lab that did a lot of work in this project. The collaborators that we had for 2Photon and for their advice, and all of you for attendance and for your attention. Thank you.
Thank you so much, Ermioni, for your great presentation. We will start the discussion session from now on. So people who have a question can unmute yourself and start your video. But let's start from the questions already existing in the chat box. So we have a question from Philip Eskenays. So Philip, would you like to ask by unmuting yourself directly, or do you want me to read it through? Or maybe he's not here. So his question was, do microglia separate to M1 and M2 or other displays? So Ermioni, could you answer to that question?
Yes, we have not tested for M1 and M2 markers, but they seem to acquire pro-inflammatory state, type 1 interferon, and MHC2 along with CD86 are usually more of an M1 type of microglia, if I could say what microglia is leaning to, but we haven't checked for the specific markers that defines M1 and M2 microglia.
Okay, thank you. And the second question in the chat box is from Ellie Huizera. So Ellie, are you in this room?
Yes, can you hear me?
Yes, I can hear you.
Basically, my question was I guess in regard to your EV isolation and just like how do you make sure your EVs are just microglia specific with no contamination from other types of brain EVs and so forth.
Yes, thank you for the question. We cannot exclude that they are derived from other cells, but we see an enrichment in CD11B levels in knockout mice. We also see some in some stainings I haven't shown you, but are in the paper, a higher biogenesis of EVs, higher staining of CD63 and CD9 marker, EV marker from microglia. So we presume they secrete more. And that's, of course, a good point. And we would like to further purify this population by using beads that are labeled with CD11B or a marker suitable for microglia. If you're asking how to make this more specific, of course, there are some EVs from other cells as well when we isolate from brain parenchyma and from the cerebrospinal fluid.
Oh, okay, thank you. Just kind of like a quick follow-up. Have you maybe you might mention it? Have you tested to see whether the microglia derived EVs have high expression of CX3CR1? I know it's a very popular marker for microglia.
No, we haven't tested that. We have just tested CD11B in Western blots.
Okay, all right, thank you.
Thank you.
Okay. And the next question is from Frederick and Naftalin. Frederick, are you there?
Yes.
Yeah, you can feel free to ask your question.
My question has to do with the generality of your findings. The microglia come from two sources. I would say generally it's accepted that about half of them arise in the CNS de novo, and the other half are monocytes that traverse the blood-brain barrier and become microglia. So my question is whether exosomes from systemic systemic monocytes or macrophages might have the same effect, or conversely, whether you could expect similar effects in systemic organs from circulating monocytes or macrophages.
So it would be interesting to test if other organs are affected as well. We haven't tested, we haven't done experiments to test the input from monocytes outside the blood-brain barrier. We know we don't have monocyte infiltration, but EVs, of course, can cross. What we have seen in mice that are bone marrow and have a defect in ERCC1 in bone marrow derived macrophages, they reprogram cells metabolically. So they make tissues uptake more glucose and enhance inflammation. So this is another mouse model. It's not in this one, but we haven't tested anything from the monocytes in the brain.
Thank you.
Thank you.
Good. Thank you, Frederick. Is there anyone having questions to Ermioni?
So I have a question. Very nice talk. I have two questions. One is why is this phenotype relatively robust in cerebellum instead of other brain regions?
So you mean if microglia is different? We haven't done something to test if microglia, the phenotype of microglia is different or more pronounced in terms of migration, branching. I mean, you see branch cells in cerebellum and spinal cords as well, but we haven't tested, we haven't really compared to see if they would be more specialized in one of those functions that you see if they produce more EVs or if they communicate in different ways, because I know microglia are they usually acquire different characteristics in different brain regions. The microenvironment plays a very important role, and when you talk about chemokines and signaling, of course, that's also very important. It could be an interesting follow-up to see if cerebellum microglia, for example, is a bit different.
Yeah, my question, do you see any also some tunnel staining signal in other brain region, which is enriched for microglia, such as hippocampus or some regions, or substantia nigra region, which is not also has microglia population?
So we have mainly focused on the cortex that we did not see differences, but I do not remember seeing in the hippocampus higher percentages of cells that are dying, at least from tunnel staining.
Thank you. A second question, you treated EB with a DNS1. Do you think that the DNA is mostly outside the EBs, or the DNS can also, clear the damaged DNA in the EB as well in your experiment?
So you mean how DNA is in the EVs, or what?
Yeah, so you treated the EB with DNS1, right? So in that experiment, I guess if you damaged DNA is outside the EB, it can be digested by DNS, but if it's inside the EB, I think it's a little difficult. So I was wondering how much of the damaged DNA can be, digested by DNS treatment. So did you
DNA
see that? Yeah, that's
DNA
my
is
question.
studied with use of saponin, which is a detergent. So it's inside the EVs, and EVs then fuse with microglial membrane. So it gets inside microglia cells, and we presume that they target the DNA fragments there mostly, but also I can imagine they would fuse with other EVs and digest the DNA. But you mean the DNA that is on the outside of the targeted EVs? That could be. I don't know if that. I don't think that could be targeted at the same at the time of
So
fusion.
I see. So you treat the BB with saponin, which is a membrane permeable reagent. So it makes sense. Yeah. Okay. Thank you.
Okay. And there is another question in the chat box from the Frederick. So Frederick, can you ask the question?
The inflammatory response related to macrophages and microglia is strongly affected by hormones, particularly estrogen. And as a first approximation of asking this question would be to look at whether there are sex differences in the effect of the exosomes, or even of the exosomes themselves. Are they different between males and females?
So we haven't really separated between males and females, but that sounds like an interesting point. We haven't observed differences in terms of neurodegeneration, I mean paralysis, but we haven't tested at the level of tissue or culture to see if
Thank you.
it wouldn't be as activated, for example. We haven't.
Yeah, it would be a relatively interesting and straightforward thing to separate the sexes.
Yeah, sure.